Okay, in this video, you are going to learn regulation of enzyme activity. When reaction occurs and at what rate. All living organisms need enzymes to function properly. You know that enzymes catalyze many chemical reactions without being consumed in the process. But what controls enzymes? There will be chemical chaos if all the metabolic pathway operates simultaneously. A regulatory mechanism involves controlling the enzymes produced, control when and where these enzymes are active. Let's take a look here. Genes that codes for enzyme may be turned on or off. Let's say if the genes are turned on, then the enzyme will be synthesized. For example here, gene that codes for beta-galactosidase, this is enzyme, is activated when lactose is present. Another example, inactive enzyme, such as this pepsinogen. Pepsinogen contain extra amino acid sequence that inhibits enzymatic activity. It is a zymogen, inactive precursor of pepsin, the principal proteolytic enzyme in gastric juice. Next, presence of inhibitor. There can be reversible inhibition. Next, regulator molecule may activate or inhibit enzyme, such as allosteric regulator. This one we will cover in the next slides. Next, end product may inhibit enzyme, such as in feedback inhibition. Allosteric regulation of enzymes. Regulatory molecules bind briefly and reversibly to a site other than the active site, or this is the allosteric site, and change the shape and function of the active site. This allosteric regulation can be divided into two. It can be either allosteric activation or allosteric inhibition. This allosteric activation happens when the allosteric regulator binds to the enzyme, thus activating it, while the allosteric inhibition is a form of non-competitive inhibition. Allosteric regulator binds to enzyme, inactivating it. This diagram shows allosteric regulation of enzyme activity. Most enzymes known to be allosterically regulated. They are constructed from two or more subunits, such as this one you can see, 1, 2, 3, 4 subunits, each composed of a polypeptide chain with its own active site. Here are the active sites. The entire complex oscillates between two different shapes. Here, catalytically active, and another one, inactive. The binding of an activator to a regulatory site or the allosteric site stabilizes the shape that has functional active sites. So here, it stabilizes the active form. But then, when there's a binding of inhibitor, it will stabilize the inactive form of the enzyme. A shape change in one subunit is transmitted to all others. Through this interaction of subunits, a single activator or inhibitor molecule that binds to one regulatory site will affect the active sites of all subunits. Fluctuating concentrations of regulators can cause sophisticated pattern of response in the activity cellular enzymes. At low concentration, activators and inhibitors dissociate from the enzyme, and the enzyme can oscillate again. This is another type of allosteric activation. These mechanisms are known as cooperativity. Here you can see the inactive form shown on the left oscillates with the active form when the active form is not stabilized by substrates. This mechanism amplifies the response of enzymes to substrate. You see here, when this substrate binds to one of the active sites, it will cause the second and subsequent substrates to bind more readily. Again, I repeat here, the binding of one substrate molecule to active site of one subunit locks all subunits in active conformation. Metabolic pathways are a series of reactions catalyzed by multiple enzymes. Feedback inhibition is a mode of metabolic control in which a metabolic pathway is halted by the inhibitory binding of its end product to an enzyme that acts early in the pathway. The end product later will become the inhibitor. It will bind to the allosteric site, and it is an allosteric inhibition. Example is the accumulation of isoleucine allosterically inhibit the enzyme in the first step of the pathway. What is the importance? Okay, it will prevent the cell from wasting chemical resources to synthesize more isoleucine than necessary. So this will avoid wasting chemical resources. This diagram shows feedback inhibition in isoleucine synthesis. Bacteria synthesize the amino acid isoleucine from the amino acid threonine. The isoleucine pathway involves 5 steps, each catalyzed by different enzymes. You can see here enzyme 1, 2, 3, 4, 5. When enough isoleucine accumulates in the cell, the isoleucine inhibits threonine deaminase, the enzyme that catalyzes the first step in this pathway. You see here, this threonine, it is the initial substrate. It binds to the active site of enzyme 1, the threonine deaminase. This is the metabolic pathway. The end product is isoleucine. If enough isoleucine is present, or when there's accumulation of isoleucine, it slows down its own synthesis by allosterically inhibiting the enzyme for the first step of the pathway. Here you can see this isoleucine will bind to the allosteric site of the first enzyme, which is the threonine deaminase. You can see this active site is no longer available and this pathway is halted. The binding to the allosteric site is reversible. When the isoleucine concentration is low, threonine deaminase becomes active. Don't forget to like and subscribe our channel. Thank you.